All right, let's see what you said. For if you have those two transposons, can that affect transcription? And most of you said yes. So let's go by parts. Okay, you were right if you said yes, because often if you say, can this happen? Almost always in biology, yes, it can. No, it's like, is it likely? Then it's like, because biology is not very deterministic, which drives mathematicians crazy, or at least my husband, because he likes, this is the rule, things follow the rule. But in biology, it's like, actually. So, all right. You could say maybe Dr. T says that it can affect because we already know that a transposon in the promoter can affect transcription of this gene. And by affecting transcription, then it can affect translation, as in how many mRNAs do you have to make this protein? So the amount of protein made may be lower if you have less or fewer copies of mRNA. So in the next question, it's like, how about if you have these two? And then like, well, we already know that this can affect, then probably, no, this can affect too. But like, okay, I guess the better question would be, does this add to the effect of this? And now we're going to see the answers to that. Okay, yes, it can. Okay, so here you can see a picture of the activity of the chalcone synthase, and it's tissue specific. For some reason, this part of the flower, it doesn't, there is no transcription, and then in here there is. So that has to do also with the transcription factors, but that is a future slide. So what's the point of this here? This is to say that when you have a transposon in the regulatory sequence, then the amount of expression is 1/7 of when you don't have that transposon. And the reason for that is that this DNA sequence here, you can think about it as like, what's it called? A fly paper. Anyway, imagine, like, I don't know if you guys know this. There's like these yellow sticky papers that you, in agriculture, you might put near your garden so that it attracts the flies, or the bugs, so they don't go on your plant, they go on the yellow thing. That could be like, here is the DNA sequence that attracts proteins called transcription factors. And then if this sequence is changed, then the transcription factors don't bind as well. And then if the transcription factors, which are other proteins, so there has to be a bunch of proteins that bind here in the regulatory sequence and the RNA polymerase to get this transcription. If this changes, the transcription factors don't bind or don't bind as well. And so you have fewer mRNA molecules made out of this. So a little bit fewer. You're like, whoa, that decreased a lot. And it's the presence of this other transposon. And you're like, my goodness, but what is it? What's up with this? Like, it's downstream from the gene. We know that this region upstream of the gene regulates transcription. How come something downstream decreased it so much? And like, oh, this is totally white. And then something I didn't tell you earlier, but I'll reveal to you now. See these arrows here? This is the direction of this transposon sequence. So this is downstream, going down, and this is going up. What that means is that this and this are complementary, and they are both the TEM3 transposon. So, but the TEM3 inserted itself in this direction here, and this other one inserted in this direction here. What happens is that this glues to this, and they make a loop, and by making this loop, then the transcription factors cannot access the promoter. You're like, my God, this craziness. Yeah, let me show you figures of that so that you can see that. Okay, I wanted to point out that the gene sequence is the same in all three, right? So the exons are here all the same in all three versions. The introns are the same. The 3' UTR, the 5' UTR, in this case, they're all the same. What changes is the presence of the transposon in the regulatory sequence or downstream from the gene. So this is an example of the gene itself. The RNA that's made out of this gene is the same, but how much mRNA gets made is what causes these different phenotypes. So I told you the thing about this loop-de-loop. Let me show you the model that people proposed. Here's the Nivea gene. That's the one that encodes that enzyme, the functional enzyme. And if you have a TEM3 in the promoter, these are the transcription factors. They bind, but they would bind better if this TEM3 wasn't here. But in the presence of TEM3, you still get some transcription, and that's how we get this color here. But when you have the two transposons that are inverted, they bind each other, form this loop, and here are the transcription factors. They're not able to bind the promoter to then transcribe this gene. So in molecular biology, let me tell you some of the things. This little arrow, it's really tiny here, but an arrow means this happens. So this process happens. When the arrow has like a on the bottom, that means this is not happening. This binding is prevented, or it doesn't happen. Okay, so do we have to memorize any of this? No. What I wanted to know is that the level of transcription of a gene, so the gene itself may be the same, but how much it gets transcribed influences the phenotype. And how much it gets transcribed can be with messing with the promoter sequence. And even in this last case, you're still messing with the promoter sequence, but because of this folding. So this is a specific case to show you that even some transposon that is not in the promoter can affect gene transcription.